Good morning to every one of you. Yes, and welcome to the final and last day of our online program, introductory course to Microbiology and Biotechnology, offered by the Department of Microbiology and Biotechnology, Sister Nivedita University, Kolkata. This is our last and final day. So actually, from tomorrow morning, I think we all will miss this session in the morning to get early and to brush it up and to complete the breakfast and get ready and sit inside in front of the computer. We had a webinar. So this thing we are going to miss from tomorrow, but hope that very soon we will come up with another program, online program, as we all are going through this new normal situation. So probably we will be coming back very soon with another this kind of interesting program. And today, the last day, we have another very interesting talk. I mean, two very interesting talks. And I'm very happy to inform you that one of the speakers of today is coming from abroad. Okay, he is. We have now our webinar has come to an international level actually. So, from our department, Dr. Milon Banik will be giving you a lecture, and from the SciTech Biosciences, Dr. Edmond Chua will give us a lecture today. He is a sales and marketing manager, SciTech Biosciences, East Asian region. So, he will be speaking on the introduction to spectrum flow cytometry, and Dr. Milon Boni, he is going to be, talk today on the basics of and principles of genetics. His topic is from gene to genetics, a journey to the inheritance biology. So these two lectures we are going to have today, and I'm very much excited to listen from them, to learn from them. I think you all are very much eagerly waiting for them. May I know that Dr. Bonik, have you joined? Dr. Bonik,
Yes,
please.
Dr. Paul, I can hear you. Good
Okay.
morning, sir.
Good morning. Good morning. So, just it is 10 a.m. and we are going on our schedule. So, without wasting any much time, each and every day I am speaking so many things, but today is the last day. So, same thing I'm not going to repeat. I'm just going to introduce Dr. Milan Boni. He is a very, very talented faculty member of our department, very dynamic and very research oriented personality. He is very much enthusiastic, very good teacher, and very good researcher. Okay, he has done his PhD from Kalyani University from the biophysics department, and after that he has done his postdoctoral research from the Indian Institute of Chemical Biology, CSIR Kolkata, and after that he has joined our department as an assistant professor, and now he is working and teaching, mainly focusing on the genetics and nanoparticle research. So without wasting much more time, I'll invite Dr. Boni to deliver his lecture. So I'm inviting Dr. Boni. The screen is yours, as I always say to every speaker that we cannot say that the stage is yours in this virtual platform. I must say that the screen is yours. So Dr. Boni, please, I'm inviting you for your tremendously good lecture. Dr. Boni.
Uh, Sir, can you hear me?
Yes,
Hello.
yes, Dr. Boni. Yes,
Oh,
yes.
okay. So, thank you, Rose sir, and thank you all of my colleagues for giving me the option to sharing my some sorts of lecture in this virtual ground. And I also contribute my thanks to all the viewers, listeners who also keep their passion to day by day to hear this kind of enlightening lectures. So, may I just 1 minute? Rajat sir, is it shared? Hello.
Yes, Milon sir, it is shared. We can see this.
Okay, okay. So,
Okay.
as Rajat sir asked me to talk something regarding very interesting regarding genetics. So I focused rather than a talk, rather than a journey. So I gave my lecture talk from gene to genetics, a journey to the inheritance biology. So it's a very journey rather than a talk. So we will explore in this coming 1 hour how genetics evolves, how the science related to genetics have been emerged, has been emerged. So first of all, what is gene? There is a historical insight. So gene is not discovered as I think now. So Mendel first discovered the law of inheritance. So he is uh, regarded as the father of genetics or modern genetics. Not only so, but from Aristotle time, there are so many things regarding genetic or inheritance biology. So after Mendel discovery, around 50 years, I will tell about how Mendel discovered how he formulated his work. So after Mendel discovered, it took another 50 years for three scientists, Hugo de Vries, Carl Corens, and Erich Schermer. They are from different Western European country. One is Hugo de Vries from Netherlands, and Karl Corens and Erich Schermer from Germany and Austria. They re-hypothesis the same thing what Mendel did 50 years ago, around 1890. After that, Walton, Shudon, and Theodore Bovary, they independently developed different parts of chromosome of animal or plant cells. They develop independently and propose another theory of chromosomal theory of inheritance. So this is a back-to-back discovery. So, just 1 year later, after Walter Schutton, Theodore Boveri also proposed same kind of approach, and the theory is collectively known as Walter Boveri-Schutton theory of chromosomal inheritance. And after another 7 years, the term gene was first coined by Joe W. Johansson. So, at previous to 1909, uh, the genetic marker, favorably known as a factor that contains trait or that is responsible for a particular trait. So, after 1909, Johansson first coined the term gene, and just 1 year before, uh, year Garrod just proposed one gene, one product hypothesis. Actually, he formulated this hypothesis from some model animal rather than some biochemical analysis. So we'll see later that some another scientist will discover one gene, one enzyme hypothesis from biochemical or molecular biological approach. And then the wonderful discovery that was performed by T.H. Morgan in 1911, he proposed gene association or gene association with chromosome, genetic linkage, etc. This is a very beautiful discovery in the field of science, in the field of genetics, and it took another almost 30 or 40 years. There is no huge discovery related to direct field of genetics. L. Pauling and Ingram, 1949, established the role of gene in protein synthesis. Now, the molecular biological approach of gene has been established by the work of Pauling and Ingram. Say, propose the gene can be expressed, and that is responsible for the expression of protein. So, gene, how it performs in our body through protein has been elaborated by their work. At the same contemporary, Biddle and Totham proposed one gene, one enzyme hypothesis. This is completely molecular biology or biochemical approach rather than the previous approach. And we know better that after 4 years, 1943, Watson and Crick proposed double helical model of DNA. They are very famous scientists. They marked a new entry in the field of molecular biology, in the field of genetics. So they proposed double helical model of DNA.
Milon sir, we cannot hear you.
Hello.
Yes, now can you repeat from the Watson and Crick 1953?
Okay. So, around 1953, Watson and Crick proposed double helical model of DNA. So, this discovery marked a tremendous revolution in the field of genetics, in the field of molecular biology. And after 3 years later, the fine structure of gene was proposed by Seymour Benzier. So it is one of the tremendous discovery. And 1960, Barbara McClintock also proposed the mobile genetic element, so mobile element of DNA that is jumping from one portion of chromosome to the another portion of the chromosome that is preferably known as transposon. So now another Janowski, 1965, also supported one gene, one cystone or one gene, one polypeptide hypothesis. So this is also this supporting discovery with the previous discovery and 1975. Now Frederick Sanger, who discovered the method for DNA sequencing. So last uh, 1 century, last 100 years, there is no sequence of the DNA. But Frederick Sanger developed the technique how we can sequence the DNA, how we can sequence the gene. So unless until you can sequence the DNA, you cannot able to see the pattern of DNA or pattern of gene is related to protein expression and etc. So back to the discovery carried out by Maxim Gilbert. They are from America. They also published a different technique for DNA sequencing. And 1984, this is one of the revolutionary discovery of molecular biology or genetics and cell biology. Carey Mullis also discovered PCR. So you see that Sanger developed the technique how we can sequence the DNA or gene, and Carey Mullis also developed the technique how we can amplify DNA or gene in vitro, in a test tube, in laboratory. So this is a complementary development. And 1996, the dolly was first female domestic sheep. This is a cloned mammal that has been introduced in 1996. So this was a remarkable achievement in genetics, in molecular biology, and the complete genome project. So human complete genome. So sequence of total chromosome is also observed or also published in 2009, 2003. So just as I told you earlier that it is a journey rather than it's a talk. So I emphasize some main topics. So first we want to introduce what is gene, then classical genetics that is related with more Mendelian genetics, extension to the Mendelian genetics, genetic linkage, genetic disorder, and non-Mendelian inheritance. And ultimately, the final talk would be sequencing of gene and genetic engineering. So this is the modern or current views of genetics and current application of genetics. So classical definition of gene. So first of all, now we see that gene, it took another 50 years to define the nature of gene. So in the before 20th century or at the last moment of 19th century, there were different kind of arguments regarding definition of gene. Some are called they are recon, some called they are mutant, some called they are system, some called they are compron. So what do you mean by recon? What do you mean by recon? So at that moment, that is early 20th century, some scientists proposed the smallest fragment of DNA that can capable of undergo recombination. This smallest fragment of DNA is better known as recon. And this is also true for mutons. The theory who proposed muton, they also suggested that muton is the smallest unit of DNA that can undergo mutation, that is changes the DNA structure and changes the DNA structure and
Milon, sir? We cannot hear you.
Okay, can you hear
Yes.
me?
Yes,
Yes, yes.
sometimes the net maybe get disconnected.
Okay,
Okay.
may I start? Can you see my?
Yeah, your
Mute
slides are
on.
visible.
Yeah, mute on. From mute on, you just please repeat Milon, sir.
Oh, sorry, sorry. Actually, a muton is the smallest unit of DNA which can undergo mutation. Muton officially means the smallest fragment of DNA that will undergo mutation, and it after that it will show a visible effect in the animal in the bacteria. The recon and muton concept is mainly concentrated on bacteria and plant biology because they are supplemented by botanists. Cistron and complon is another definition of gene. Cistron means this is correlated with the functional approach of gene. So functional approach of gene means the gene is one gene is one protein or one gene is one enzyme. So cistron refers to this kind of function or related to this kind of function. Complon. This is the unit of complementation. This definition was derived from bacteriological study. Suppose some fragment of gene can complement the lack of activity of bacterial cell. So, in this context, the function of gene or the definition of gene was coined as complon. This is the current views. After 20th century or after the mid of 20th century, the current views of gene had been published. So this is structural gene, inducible gene, operator gene, and regulator gene. Structural genes means this is the housekeeping gene. They are always expressed. They are always expressed into mRNA that will undergo translation. So I think Dr. Thakur Chaudhary, day before yesterday, emphasized the molecular biological approach related to gene expression or translation. An inducible gene is another form of gene. They are also present in our body, but they are not always expressed or they are not always functional. So the third type of gene is operator gene. So operator gene balance the expression level of inducible genes and structural genes. So when and in which circumstances inducible gene is expressed or is not expressed, that is coordinated by operator gene. And the final type of gene is regulator gene. So regulator gene will synthesize some protein or some factor that will bind to the operator gene and ultimately finally regulate the function of operator gene. So this is the current views or current function of gene. One is structural gene, another is inducible gene, another is operator gene, and finally regulator gene. So this is the famous experiment. That was the experiment that gave birth to the modern genetics. Though we know this is the Mendel experiment. He did the experiment in sweet pea. Actually, there are so many variants of sweet pea. I try to categorize the variant according to their height, flower color, flower position, seed shape, pot shape. Mendel chose sweet pea. There were some reason because uh, due to self pollination or cross pollination, they also choose character with only two states or phenotypes or created pure breed lines. This is very important. Created pure breed lines means suppose he choose tall character of a sweet pea. So he also produced ultimately tall plant from this tall plant, and he termed this tall character is a pure. This is also true for all the remaining variant or all the remaining character. Actually, what he did, he just took two flowers from same plant, from different plant, and removed the genital part or a male part and introduced physically the pollen into the anther and perform the artificial, I want to say artificial cross pollination. And ultimately, the plant all the newborn plant are tall in nature, but when he crossed this tall variety into in the F2 generation, there is a huge difference. The dwarf property or dwarf variant was also emerged in the F2 generation. So there is a two population. One is tall and another is dwarf, and the ratio of tall and dwarf is 3:1. So we know this renowned experiment. So to analyze this experiment, he introduced two terms. One is genotype and another is phenotype. So genotype is the genetic profile of an organism, and phenotype is the phenotypical, that is character that is expressed and that is what is visible of an organism. So he termed the tall plant as a capital T, small plant as a small t, and the hybrid plant that genotypically is capital T small t, but phenotypically small genotypically is capital T small t, but phenotypically they are tall plant. They were tall plant. So what Mendel observed from these seven types of variant, tall plant against dwarf plants, round sheet against wrinkled sheet, the he just marked the ratio, and you can see the ratio belongs to 3:1 in F2 progeny, that is the second generation, not F2 progeny. So he marked a new definition. So whenever you cross a single trait in first generation, you will only achieve only the dominant property, and when you let them to cross in the another next generation, dominant property and recessive property will be explored, and the genotypic ratio would be 3, a phenotypic ratio would be 3:1, and genotypic ratio would be 1:2:1. So Mendel postulated his theory into some comments rather than a few points he tried to explore. So there are hereditary particles that determine traits or character. That is gene. So genes are in pairs, that is preferably known as alleles. So I told you earlier that Mendel marked the term gene or as a factor, but now it is favorably known as gene, so I introduced gene in place of factor. So there is a pair of gene, so each gamete carries only one allele, and that's all. So another thing is homozygous and heterozygous. Homozygous means a single progeny has one pair of chromosome for each allele. So in case of homozygous, they contain same type of allele. In case of heterozygous, each chromosome has different kind of allele. So dominant allele and recessive allele. As I stated earlier, dominant allele which is expressed both in homozygous and heterozygous condition, recessive allele which is expressed only in homozygous condition. Am I audible? Rose sir? Hello.
Yes, sir, you are audible.
Okay, sir. Thank you, sir.
Hmm.
So he introduced two terms. One is dominant allele, another is recessive allele. Dominant allele means which is expressed both in homozygous and heterozygous condition, and recessive allele which is expressed only in homozygous condition. And Mendel summarized his formula that is preferably known as law of segregation. I present the formula in my slide that states the two copies of a gene segregate from each other during transmission from parent to offspring. Therefore, only one copy of gene is found in a gamete. So, only out of two allele, only one allele will be transferred from parent to offspring, and when it will form in F1 generation, each one gene will each copy of one copy of each gene is found in that particular gamete. That's why in case of F1 generation, we will observe the genetic nature is capital T small t, whereas in case of F2 generation, we will observe the genetic nature is capital T small t. So Mendel summarized his formula that is preferably known as law of segregation. seed and as well as the color of the seed. So yellow and round shape versus green and wrinkled. You can see the nature of color of the seed as well as its shape versus green shape wrinkle green color wrinkle shape varieties. So when he introduced this same kind of uh, cross in F1 generation, all the progeny were yellow round, and when yellow round generation were crossed for the F2 generation, the different varieties will be observed, and he marked in the graph. So there are four types of plants he observed in the F2 generation. One is yellow round, another is green round, another is yellow wrinkled, another is green wrinkled. So yellow round is a mother progeny like green wrinkled. Green round and yellow wrinkle is the recombinant progeny. Though he did not mark the term of recombination, he just find out the ratio and he observed the genotypic ratio as well as phenotypic ratio. The observed phenotypic ratio is 9 is to 3 is to 3 is to 1. Out of four different varieties, the phenotypic ratio was 9 is to 3 is to 3 is to 1. This is very important information. So whenever we proceed the next experiment conducted by different groups of scientists, they try to evaluate or try to match the phenotypic ratio in F2 generation along with this 9 is to 3 is to 3 is to 1. So from this dihybrid cross, he introduced another formula. This is known as independent assortment steps. So in this formula, he explained two different genes will randomly assort their alleles during the formation of haploid cells. So in parental generation, he took two varieties, that is color and shape of the seed. In F1 generation, only dominant property he observed, and in the F2 generation, the both property had been segregated. So if the both property have not had not segregated, uh, in case of F1 generation, we will observe this kind of nature of gene segregation. But he observed, I want to say the right portion of my slide, as the gene is independently assort, they are deviate from one another during gamete formation of in the haploid cells. So the haploid gametes in F1 generation there were four types: RY, capital Y, capital R, small y, small r, capital Y, small r, small y. If they were not segregated independently, we will get only two types of allele. That is 50% of capital R, capital Y, and 50% of small r, small y. So he, from this dihybrid cross, he supported a new experiment, a new formula, the law of independent assortment. What this formula stated? In this formula, he tried to conceptualize if you keep two different traits in a organism and let them to cross in F1 or F2 successive generation, the two marker or two character will be independently assort. They will not be relatedly assort from one generation to the next generation. So now I want to focus deviation of Mendel hypothesis. So dominance is not universal. So Mendel proposed recessive relationship, dominant relationship. Dominant, which is expressed both in homozygous condition and heterozygous condition. Recessive allele, which is only expressed in homozygous condition rather than heterozygous condition. But this dominance is not ultimate or is not universal. There is so many variation in combination of homozygous give insight in details each and every type of deviation. So first of all, hello, may I audible? Hello,
Yes, sir, you are.
Yeah,
hello.
now you are audible, but meanwhile, sometimes I mean you are not audible. Maybe your net problem. Okay, sir, carry on, please. Now it's
Oh,
audible. Yes.
so I want to say there are so many types of deviation of Mendel hypothesis. So one is partial dominance. In partial dominance, I just try to focus my figure. So in you see in parental generation, that is red follower of P, and another is white flower of P, and when you cross in you should get red flower or either white flower, but you observe red flower. So this is partial dominance. Actually, in this observation, you just find some intermediate color. When you perform experiment of red P with respect red snapdragon flower with white snapdragon flower, if in F1 generation you observe pink color of a snapdragon flower. So in that case, in F1 generation, you will observe intermediate color rather than any of the parental color. Rather than I want to say, you observe pink color in place of red or white. This kind of dominance is better known as partial dominance. And when you cross this pink flower into F2 generation, out of four flower, you will get one red flower, one white flower, and two pink flower. So in the F2 generation, all the gene had been segregated as stated by Mendel. So this is partial dominance. Partial dominance means, so in the F1 generation or in the heterozygous condition, only intermediate nature of the character will be expressed. This is one deviation of partial dominance, and this is the molecular biological this is the genetic approach. So I just skip this slide. Another is co-dominance. We learn about partial dominance. In case of partial dominance, we will observe intermediate nature of the phenomena in F1 generation or in heterozygous condition. But in case of co-dominance, we will observe both type of phenomena. Suppose the best example is our blood group. So we have four types of blood group: A blood A type, blood B type, blood AB type, and O type. In case of AB type, both type of phenotype will be expressed. I just go to my next slide to explain this phenomena. Actually, blood typing is uh, has been carried out based of their cell, glucose presence of glucose moiety on the red blood cell surface. So according to glucose moiety, blood grouping has been performed. There are four types of blood group: blood group A, blood group B, blood group CA, blood group O, and blood group AB. And in case of blood group AB, you will observe both type of antigen. One antigen is assigned for blood group A and another antigen is assigned for blood group B. So in case of AB type of person, in AB blood group, all the red blood cells display both type of antigen rather than only single type of antigen. So A antigen and B antigen are not dominant and recessive for each other. They will be expressed in the single type of red blood cell. So they are co-expressed. So this is favorably known as co-dominance. So co-dominance is another form of deviation of Mendel's statement. In this co-dominance, both the character will be expressed. So difference between partial dominance and with respect to proper dominance with respect to co-dominance is that in case of co-dominance, both allele will be active and both antigen or both phenotype will be expressed in heterozygous condition. So this is one deviation of partial dominance. Another deviation of partial dominance is co-dominance. So different types of allele rather than two alleles, as stated by Mendel, will take part the formation of coat color. Mendel focused on presence of two alleles for a single gene. But if you observe the coat color of rabbit, the coat color, there is a huge variation of coat color. There is a Himalayan coat color, food coat color, chinchilla coat color, and albino coat color. And the actual reason for these different varieties of coat color is due to the presence of more than two alleles. There are multiple alleles take part in this coat color formation. So, hello, may I audible? Hello,
Yes, now you're audible.
okay. So there may be some interruption. Hello, hello.
Yes, MB sir, you are audible.
Okay, okay. Thank you, sir. So, actually, in case of Mendel hypothesis, what we observe for a single phenomena or for a single gene, there are two alleles. But this theory cannot support when we try to explain the coat color of rabbit. So, scientists observe more than two alleles will perform their activity for this coat color of rabbit. So they introduce there may be some option or there may be some genetic background when there are more allele will be responsible for a single type of character. This is one type of deviation of Mendel hypothesis. So now we introduce the major deviation of Mendel hypothesis, that is gene interaction. In Mendel's phenomena, Mendel did not observe any gene-gene interaction. That means one gene will be expressed without the involvement of another gene. Gene interaction is a new terminology. It refers when one expression of one gene is under balance of action of another gene. So there are one pair of gene or involvement of more than two genes to indicate a visible phenomena. There are different types of gene interaction that may be epistasis, that may be complementation. As I am running late and we have limited times, so I just explain one single type of gene interaction, that is epistasis, in my next slide. So just we introduce in my slide, there are two wide variety of P. White variety, one white variety, this is pure. Another second white variety, this is also pure. But when they undergo a second type, if cross pollination in F1 generation, we will see a purple color formation of the sweet pea tree. So try to understand. You started your work from one white variety of pea. You started your work another white variety of pea, but in F1 generation, the outcome should be white, but the outcome should be purple in place of white. That is the mother color of the pea mother progeny. So when you observe second type of generation, that is F2 generation, when you cross this purple plant, you observe the huge variation. The genotypic ratio, I want to say a phenotypic ratio, rather than 9 is to 3 is to 3 is to 1, you get 9 is to 3, the phenotypic ratio. So actually you observe 9 is to 3 in place of 9 is to 7 in place of 9 is to 3 is to 3 is to 1. This is due to the involvement or interaction between two genes that take part color formation. This interaction between two genes is better known as epistasis. Actually, epistasis defined when one gene deregulates the expression of another gene. So one gene will negatively regulate the expression of the second gene. This is known as epistasis. So epistasis, the mother color of the pea tree, is purple in place of white. So epistasis defined when one gene deregulates the expression of another gene. So gene linkage. So I want to. This is another. This is completely different topics. What Mendel observed in his classical experiment. So in gene linkage means when two genes are linked with each other. Actually, Mendel performed all the classical experiment with seven types of varieties of sweet pea, but luckily, he never faced the problem of gene linkage. Around 50 years later, Bateson and performed the same experiment with sweet pea, but he took to different trait rather than the traditional trait to validate Mendel's hypothesis. They took red flower versus white flower. This is one pair of trait, and the remaining pair of trait is length of the pollen grain. So long pollen grain, short pollen grain. So they also perform the dihybrid cross, but without taking the similar type of character that Mendel had started with. So they took red flower long pollen grain with respect to white flower short pollen grain. So in F1 generation, they observe all the flower are red flower long pollen grain. But when this F1 generation were interbreed among themselves, the F2 generation there is a huge variation. Expected ratio should be 9 is to 3 is to 3 is to 1, but the obtained ratio is 24.3 is to 1 is to 1.7 is to 1. So according to Mendel hypothesis, the ratio should be 9 is to 3 is to 3 is to 1, but they observed the peculiar relation in F2 generation, that is 24.1 is to 1 is to 1.7 is to 1. This observation totally different from Mendel's observation. They repeated the same experiment with taking different variation of flower color with respect to the length of the pollen. And they also observed the same type of observation, and that is totally different from Mendel observation. That is well-known observation in dihybrid cross, that is 9 is to 3 is to 3 is to 1. I indicate, I mention every information in my table of the slide in the slide. So you can see the observed ratio, there is a huge variation with the respect to the expected ratio. So this observed and expected ratio, there is a huge variation. So their data could not support Mendel data. They try to explain this result with respect to the meiosis cell division pattern, because I just told you prior to my just told you in my initial slide, Sutton and Bovary introduced the chromosomal theory of gene or inheritance pattern. This chromosomal theory supported Mendel's hypothesis, both the law of segregation and law of independent assortment. So, just 1902, they independently discovered chromosomal pattern theory of hypothesis, and that theory also supported Mendel's formulas, that is law of segregation and law of independent assortment. They capitalize these two information. One is Mendel hypothesis, another is Sutton-Bovary's hypothesis. They just try to understand the formation of haploid cells prior to pollination or fertilization. So there are, I just try to focus on my slide, a diploid cell after meiosis, the possible haploid type. There is a two possible haploid type. In my left side, when the haploid cell had been undergone meiosis, there is a recombinant types of recombinant types of haploid cell, and if the diploid cell during formation of
Hello, Milon sir. Hello.
Hello.
Yes.
Hello.
Yes.
Sir,
Yes.
may I
Yes.
audible now?
Yes. Now you are audible.
So just I started from slide number 29 from this slide.
Slides cannot be visible, sir.
So is it visible now, sir?
No. No. No.
Uh, Hello.
Yes. We can hear your voice, but slides are not visible.
So actually, in my laptop, I just sharing my slide, but
Wait, sir. 1 second. I am sharing. Okay.
okay,
You just, stop sharing.
okay, sir. Sir, can you start from slide number 28?
Yes. Sure. I will share. Is it visible now?
Yes, sir.
Yes.
Hello. So
Is it
may
visible
I?
to all?
Yeah. So
Yes.
may I proceed, sir?
Yes. Yes. Yes.
So I think I lost my connection from this slide. So may I repeat from this?
Yes. Yes. Go ahead.
Okay. Thank you, sir. So I actually try to explain. Peterson and Punnett discovered that two assort did not assort independently. Uh, this was their experiment. They took color of the sweet pea with respect to the length of the sweet pea, and when they crossed in F1 generation and successive F2 generation, they observed the observed ratio that is is not, was not matched with the expected ratio. The expected ratio should be 9 is to 3 is to 3 is to 1, but the observed ratio is 15.6 is to 1 is to 1.4 is to 4.5. So there is a huge deviation from Mendel hypothesis with respect to their observation. So they try to explain this theory with respect to the Sutton-Bovary's chromosomal theory of uh, inheritance. Next slide, sir. So, in chromosomal theory of inheritance, Bovary and Sutton explain that there are some chances that recombinant haploid cell formation prior to gamete formation. So, when recombinant haploid cell have been formed, so in case of F1 generation and or F2 generation, there is a huge probability that two markers cannot be assorted independently. If there is some kind of recombination, two marker could not be assorted independently, what had been stated by Mendel earlier. So Peterson and Punnett just try to correlate their observation where they observed the two characters were not assorted independently with respect to the theory of chromosomal inheritance as well as the theory of Mendel's. Next slide. So actually, now the back-to-back experiment performed by Thomas Morgan. So actually, Peterson and Punnett just explain the expected ratio why it is this was the first experiment. This was the first experiment that Morgan also introduced Drosophila, and Drosophila is now used as a model organism for genetic analysis, for developmental biology, etc. So, like Bettison and Punnett, Morgan also chose two different characters. One is length of the wing, another is body color. Hello, hello.
Yes, sir. Yes, sir.
Uh, Rostar, just go to previous slide. Hello.
Yes.
Uh, uh, can you show enlarge view? It will be better for the participants.
Okay. Okay.
Okay. So there are two types of he choose two different variation. One is wing length, another is body color. He performed the experiment on Drosophila, and he also observed similar kind of deviation in F2 generation. There are four different variety of population in F2 generation. The mother variety is long wing gray body versus restal wings and black body. He observed these two type of variation along with the recombinant variation. But the recombinant variation proportion was much more lower compared to parental variation. So in that time, he introduced the term genetic linkage. Genetic linkage refers when two genes are associated very close to each other. There is a lower chances or lower probability they will assort independently during gamete formation, or that's why they were unable to segregate independently. This, this experiment marked a new dimension in classical genetics. Though this is a deviation of Morgan hypothesis, but it also indicates the powerful observation that genes are very close to each other with a little distance among them, and when the genes are very close to each other, there is a lower chances that they will segregate independently. What Mendel observed that the genes are segregated independently, or this is totally different from the Drosophila experiment or the experiment that was conducted by Bettison and Punnett in sweet pea. The next slide. So actually, not on, Mendel also performed the similar type of experiment on the sex pattern. There are two types of uh, Drosophila eye color. Now I want to introduce two different terms. One is wild, another is mutant. So what do you mean by wild? So the maximum population in nature is referred to as wild type, and different from wild type is referred to as mutant. So if you try to observe the eye color of Drosophila flies, so maximum eye color is red in nature, that is wild type, and limited number of Drosophila are mutant in nature because their eye color is white rather than red. So he introduced the term wild type and mutant. He performed the same experiment with respect to this variation of wild type with respect to the mutant Drosophila flies based on different genetic partner, that is male flies, female flies with respect to their eye color. Next slide. So Mendel chose wild type female flies with respect to white type, that is mutant mutant male flies. So he cross-breed these and mutant male flies. So in the F1 generation, he observed two different type of flies rather than a single type of flies. One is male flies, another is female flies. And when he performed this, their interbreed between these two different type of flies in F1 generation, he observed four types of flies. In four types of flies, what he observed that the mutant eye that was previously contributed by the male flies. So all the male flies, if you see, all the male flies, 50% is wild type, 50% is white color. And this is also true for the female flies in F2 generation. All the female flies is wild type in nature. So that is the genetic exchange in gamete formation did not support this experiment. Actually, the eye color of Drosophila is associated with the X chromosome. X chromosome of the Drosophila. So whenever X chromosome is segregated, two types of whenever the X chromosome is segregated, it also segregated the nature of wild type or nature of mutant gene. As wild type gene is dominant or recessive gene, so you would observe in F2 generation 50% of female flies is wild type and 50% of female flies, 50% of male flies is wild type, 50% male flies are mutant type, but 100% of female flies are wild type. This wild type gene is contributed by female flies, and in F2 generation, all the female flies are wild type. He also performed reciprocal cross, just interchange the male partner with respect to female partner, just changing their character. So in the first experiment, he chose wild type female, male type mutant male. In the reciprocal experiment, he chose just different orientation, mutant female with respect to wild type male. And in the F2 generation, what he observed? In the F2 generation, he observed all the female flies, 50% of female flies are wild type, 50% female flies are mutant. So from these two reciprocal experiment, he conclude that the gene that is responsible for the eye color of Drosophila contributed by X chromosome, that is sex chromosome. This was the first experiment sex chromosome take part in genetic inheritance pattern. And also he marked that the linkage relationship between the uh, wild genetic sex of the Drosophila with respect to their eye color. Next slide. So, uh, actually, Morgan proposed that take part, sex chromosome takes part in inheritance pattern of Drosophila. This is quite true for all the creature, all the mammals, all the reptiles. But one of Mendel's student, Bridge, also capitalize his information. Rather than crossing to different factors, Kelvin Bridge, he was a student of Morgan, introduced three different factors: body color, eye color, length of the Drosophila. Three different factors. This was favorably known as tri-hybrid cross. So tri-hybrid, Bridge performed tri-hybrid cross rather than two dihybrid cross. So the three marker was body color, eye color, and length of the wings. And he just tabulated the result. And what he observed? He observed the recombinant Drosophila had tri-hybrid cross. And that during meiosis cell division, there is a lower chances that these two genes will be segregated to each other. According to Morgan's theory, the two genes, how much they are come close to each other, there is a lower chances that they will be segregated to each other. So if we justify the different type of recombinant frequency in F2 generation or F1 generation, and we just try to explain the recombination frequency from each variation, each hybrid or each parental variation, we can justify the distance between two gene out of this three combination of gene. So he started his experiment with body color, eye color, and length of the wing, and he formulated the gene distance genetic map of these these three genes according to their recombination frequency. So Breeze and Albright, both were the student of Morgan, propose that gene frequency is matched with the recombination frequency. So gene linkage can be correlated their genetic orientation in the chromosome. So this theory also supported mathematically what Morgan proposed earlier. Next slide. Hello. Next slide, sir. So inheritance. So Morgan proposed the introduction of a sex chromosome behind inheritance pattern. So actually, there are three types of sex chromosome linked inheritance. One is sex linked, another is sex limited, and finally sex influence. Sex linked. So actually, Morgan performed all the experiment with sex linked gene. The inheritance, the gene that is solely associated with sex chromosome, these are known as sex-linked inheritance pattern gene. And sex-limited, this kind of gene is not directly is associated with sex chromosome, but their expression is depends on the nature of sex of the animal, such as milk production. Though milk production gene is contributed both the sex of our mammals, but the breastfeeding uh, phenomena is coordinated by the our holy mom throughout the civilization or developmental history. So milk production is another example of sex-limited inheritance. So this kind of phenomena is not directly associated with the sex chromosome. Rather than their expression is depend on the nature of sex of the organism. Sex influence. This is another. There is a little difference between sex limited and sex influence. Sex influence gene. This kind of phenomena is always observed, both kind of sex, but there is a huge tendency or huge probability of this kind of gene. This is known as sex influence gene expression. Sex influence gene expression is that this kind of expression you will be observed in both type of sex, but the probability of expression of particular gene is higher in one sex with respect to the another sex, such as some high blood pressure gene is coordinated with human male human being rather than female human being. So this is a sex influence genetic inheritance. So what Morgan discovered sex linkage, but inheritance pattern does not follow this common fashion. There is a more variation that is sex linked, sex limited, and sex influenced. Next slide, sir. So now, just I stated a few slide earlier rather than sex chromosome. Now I introduce variation of chromosome. You see in my slide there is a variation. There is a in the left portion of my slide there is the picture of all the human chromosome. This is known as karyotype. So you can isolate the gene and you can see the property of chromosome is fixed. So any changes in chromosome in human beings or any other organism may lead to disaster. So I just try to say how chromosome variation or how chromosome structure variation will observe during haploid cell formation. There are different types of criteria that may lead to genetic refreshment, genetic abnormality. So that may be deletion. Deletion means removal of certain gene. I mark a red arrow to indicate the portion of gene where it has been deleted. So duplication, that means the duplication, the certain portion of the chromosome. Inversion, the orientation, changing the orientation of the chromosome. Translocation, that is interchange between one portion of chromosome to the another different chromosome. The translocation means the removal or just removal of one portion of chromosome from one end and just to tag with different chromosome. And this has a also disastrous effect. And reciprocal translocation, that means the interchange of fragment of chromosome between two chromosomes, interchange of fragment of chromosome or portion of chromosome between two chromosomes. These are the all phenomena that lead to the disastrous effect, the anomalous in chromosome number, chromosome structure, and ultimately lead to the some sorts of disorder. So next slide. Uh, so actually, just I this is a common structure. So I in my previous slide I focus the the change in the chromosome structure rather than chromosome number. In this slide, I also proposed, or I also try to draw your attention how the chromosome number is getting changed during massive cell division. So there is a chiasma formation that is followed by separation of chromosome and ultimate result the formation of four different type of chromosome. But if during the separation, that is non-zinc junction, if one pair of chromosome did not separate completely, result that might result the formation of lacking of one chromosome. This is lacking of one chromosome. You see in my slide, in the left side of my figure, that is started with four chromosome, and there is a problem in after haploid cell division. In four cell, one cell get one extra chromosome and one cell lost another chromosome. So there is a probability. So one cell will get extra chromosome, one cell will lose, and one or more than one chromosome. So this kind of mucus cell division may also result the changes in also have disastrous effect in human beings and in organisms. So next slide, sir. So this is the variation of human beings. There are the incidents when we lost one pair of chromosome on when an organism lost one pair of one single chromosome, that is known as euploidy, and when it achieves extra chromosome, it is known as aneuploidy. Next slide. Next slide, sir. So now, actually, I started my these topics based on genetic disorder related with chromosomal aberration, chromosomal number. So all the genetic disorder from Morgan experiment, we know that genetic genetic behavior is transmitted from one generation to the next generation with sex chromosome, and also there are the involvement of autosome. So genetic disorder can be two types. One is dominant, another is recessive. Recessive can be also another and can be also another characteristic of known as dominant disorder. If the disorder can only be traced in homozygous condition, not in heterozygous condition, they are known as recessive genetic disorder. So in my slide, in the right side, I try to summarize type of autosomal and type of sex chromosomal disorder. Uh, Trisomy 13, that means getting extra chromosome of chromosome number 13, and there are clinical symptoms, that is mental physical deficiency, popularly known as Patau syndrome. That type of syndrome is Edward syndrome, Down syndrome, and the average changing in the chromosome number also lead to some kind of sex chromosomal disorder, that is Klinefelter syndrome, Jacob syndrome, triple X syndrome, Turner syndrome. So this you will see that getting an extra chromosome or losing an extra chromosome is in human beings is highly uh, uh, abnormal effect, and it also lead to the disease in the affected individual. So I just want to concentrate in one thing rather than explain all the autosomal or all the sex chromosomal behavior. What I want to explain in case of XXX, triple X gene. You see, in case of triple X gene, that is they are female in nature, they harbor X chromosome. So you may see, uh, ask me. So if a normal female contain two X chromosome, in case of triple X chromosome, there are three X chromosomes. So uh, it contains an extra chromosome in place of two similar type of X chromosome. But what is the disaster, or what is the its uh, toxic consequence for getting an extra X chromosome? Actually, you may correlate. Suppose you in your examination, in our examination, there are more than one question rather we have to answer. There are more than one question, then we have to answer. In the nature, though we have uh, more than though we have a pair of chromosomes, that means in each gene we have at least two allele, but the expression sorry but the expression of one allele is regulated by the expression of another allele. There are huge kind of there are the many theories behind this, preferably known as genic balance, dosage compensation. This kind of physiological phenomena regulate the expression of allele level so that the overall expression from a certain gene cannot show disastrous effect. So in case of triple X woman or triple X female, though they harbor an extra X chromosome in place of two X chromosome, but the genetic balance or dosage compensation is totally lost. So there is a disastrous effect in case of triple X women. So I want to conclude that getting an extra chromosome or losing a single chromosome is a huge effect, a huge consequence of us. Next slide, sir. So I want to focus on another sex-linked gene order. This is hemophilia. So hemophilia is a very common disease. Actually, sir, may I get another 15 to 20 minutes? Sir, hello, hello, sir.
Yes, sir. Actually, we are already going. I mean,
Late.
already we are going late. So if you
So,
can,
sir,
start.
I'll take just 10 to 15 minutes. Okay, sir. Thank you, sir. Uh, uh, Sir, can you? Okay, sir. Thank you. So another example of sex-linked disorder, we know hemophilia. The affected person is male and boy, but the carrier is mother, and the gene is because the blood clotting factor is contributed by the gene that is associated with X chromosome, and mother is associated with Y chromosome. So this Uh, s- Sir, can you hear me? Hello.
Yes, sir. Now we can hear you. You continue.
Okay, okay, sir. So just I will take another 10 to 15 minutes. Okay. So this is a sex-linked disorder. So this is a very common disorder, that is hemophilia. In case of hemophilic patient, blood fails to clot. So affected person is son or boy, and the gene that is contributed by mother. So mother can be two category: normal mother and carrier mother. But the affected person is always son. Sir, next slide. So this is the these are the clinical symptoms. The main symptom is that blood does not clot properly. So I try to skip this slide. So next slide. So genetic background, I told you earlier that the gene hemophilia, that is the gene responsible for blood clotting, is associated with X chromosome. So it is a recessive disorder. Men are more likely to get hemophilia because they are the affected person, and females have less chances to getting hemophilic behavior, and they are in they are the carrier. So this is the sex-linked recessive disorder. Next slide. So this is a Punnett square. So in this is one Punnett square. We can profiling genetic pattern of through this Punnett square, and you see that male in the F2 generation, 50% of male are normal, 50% male are affected. In in the next generation, 50% female are normal and 50% female are carrier. So nature of as the nature of genetic pattern, that the association of hemophilic factor, blood clotting factor with the X chromosome can be adjusted from this Punnett square test. So I just try to skip because I am running late. Try to do not. This is a pedigree analysis. So you see, you just go before my last slide. In that slide, we just describe you. We just explaining the Punnett square. We can analyze the nature of genetic disorder. So this kind of genetic disorder we can also analyze if you have analysis of genetic pattern of few generation, 3 or 4 generations. Suppose Queen Victoria Albert, there are the 4 generation. We have data, and we can show from this Punnett square. We can justify the nature of genetic profile each and every individual of their generation in each every generation. Suppose try to explain. Suppose I want to get the nature of Alfred, the son of Queen Victoria. So if I have the data of genetic profile of Queen Victoria Albert and his another son, from pedigree analysis, we can also justify the nature of transmittance of disease or the phenotype, such as blood clotting factor, hemophilia, from this pedigree analysis. Next slide, sir. So I now want to focus on non-Mendelian inheritance. So I want to say this is another deviation from Mendelian inheritance. In a Mendelian inheritance, or what we observe in case of Morgan pattern of genetic inheritance, we saw that sex or mark gene also take part. Both gene will be segregated if they are not linked, and if they're linked, there is inhibition of segregation. But these are non-Mendelian genetics. In this kind of genetics, Mendelian formula does not follow as this. So this is a pattern of snail coiling. So in the body of a snail, uh, the body is coiling either clockwise and anti-clockwise. If their coiling is clockwise, then known as dextral. If their body coiling is anti-clockwise, they are known as if you conduct the similar kind of reciprocal experiment, that is dextral male versus synstral female. In the F2 generation, you will observe all are dextral. So this is a peculiar kind of generation. So I do not explain in elaborately because I am running late. So I want to mark a single point. So in this figure, you can see the nature of female, the nature of mother solely contribute the nature of snail coiling. The nature of mother solely contribute the nature of snail coiling. This is totally different from Mendelian inheritance pattern. This is known as maternal effect. The next slide I will explain. I will try to explain. So next slide, please. This is the genetic background of the criteria. That's why all the newborn snail exhibit mother's nature. Actually, in mother wombs during after fertilization or embryonic stage, the mRNA will take part this coiling, and mRNA is contributed by womb egg rather than sperm. So the mRNA concentrated in the egg will take part in the snail coiling nature rather than the property contributed by sperm, that is our male partner. So this is the main reason for snail coiling, that is tend to be mothers in nature. The next slide. So extra chromosomal inheritance. This is another kind of this is another example of non-Mendelian inheritance. In this kind of inheritance, Mendelian phenomena does Mendelian's laws do not appropriately follow. So suppose there are crossing between 4 o'clock plant between there are when we are conducting this experiment according to leaf nature rather than the color of the flower. So you see the in the F2 generation, the maximum all in the F2 generation or F1 generation, all the color of the leaf contributed by the female partner rather than male partner. So all the contributed actually what I want to summarize that actually, leaf color is contributed by chloroplast. The chloroplast gene is associated in the uh, uh, egg or embryo. Mendel summarized his formula based on nuclear gene or the gene associated in the nuclear. But in this kind of inheritance pattern, we observe the inheritance pattern of gene contributed by chloroplast or mitochondria. So mitochondria or chloroplast, though they harbor extra number of genes, they also take part the inheritance pattern in the next generation, that is F1 generation or F2 generation. Due to try to skip the whole experiment. I want to just summarize that in this kind of phenomena, the chloroplast rather than main chromosome from nucleus take part in inheritance pattern. This is another deviation from Mendelian genetics. Next slide, sir. So now I want to focus some modern approach. So just in the last 50 years or 100 years, there are so many experiment, biochemical experiment, biophysical experiment, physiological genetic experiment that give a proper definition, proper physiological importance of gene. But we cannot discover the sequence of gene. 1953, Watson Crick proposed the double helical structure of gene, and Horgomindo Corona, after some years later, also proposed the genetic nature code. And our faculty, my colleague Dr. Thakur Chaudhary, emphasized the molecular biological pattern, but there are some lacking in this information, because you have so many physiological information, you have so many genetic information, you have so many physiological information, but there is there are observation in 1973, 1976, 1975, sorry. Sanger developed a method to determine sequence of the DNA or fragment of fragment of a gene. So he was a Nobel laureate. He not only discovered the how to sequence the DNA or nucleic acid, he also discovered the method so that we can sequence the protein structure or peptide structure. Also, he was one of the few scientists who was awarded twice for the prestigious Nobel Prize. So next slide, we just try to summarize. So next slide, please. This is the technique by which we can analyze the gene sequence. So it is a not so easy technique. It will take a couple of lecture to understand everything. But I just to conclude you that now we have after Sanger discovery, you have the option. So you can sequence the DNA. You can sequence the DNA, and so we get a mark a whole sequence that is coming from A or coming from G and ending A, T, G, C. So all the genetic pattern or DNA sequence you can mathematically, you can experimentally prove. So this is an outstanding discovery of modern genetic engineering field. So I skip to explain all the phenomena. So next slide, please. This was another back-to-back discovery by Maxam and Gilbert. They were American scientists. Sanger was a British scientist. Maxam and Gilbert also published chemical approach to determine the sequence of DNA. So if you can determine the sequence of DNA, you can also determine the sequence of a gene. So now we have the arms so that you can determine the sequence of a DNA or sequence of a gene. So that is really very important. So unless to determine a sequence of a gene, you cannot do a little thing in case in field of genetic engineering. So I try to skip the explanation of this slide. I just try to mention that now, just 50 years ago, due to contribution of Sanger or due to contribution of Maxim Gilbert, we got the idea how we can sequence the structure of a gene or ultimately the proper structure of proper sequence of a gene. So next slide. This was one of the fundamental discovery on the modern genetic engineering. This is better known as polymerase chain reaction or PCR. This experiment was invented by Carey Mullis. He was also a Nobel laureate American scientist. So in the last couple of slides, I try to give you an impression regarding the sequence of a DNA or sequence of a gene. Now, after the achievement of this Mullis, we can also amplify a certain or full segment of DNA or gene. So now we have two options. We can sequence the DNA and we can also sequence the gene. So you can sequence the gene. You can also amplify the gene. So now you have the lots of options. The polymerase chain reaction gives us this powerful option. So we can also synthesize chemically DNA or gene. I skip the experimental or principle of this experiment. Next slide, sir. So next slide. Okay. Now in the last couple of slides, I show now we have two options. You can sequence the DNA. You can also synthesize the DNA. So this made the background of genetic engineering. Genetic engineering means so suppose you want to create a superior creature because all the creature has some loopholes, have some drawbacks. So if you can isolate a good gene from a healthy or person or healthy individual, and if you take or inject this portion of gene into the compromised person, then the compromised person will harbor this new character, and the compromised person will be a hybrid in nature. This these are the things that genetic engineering deals. So now we have two options. We can sequence the DNA or we can amplify the DNA. So now we have the So last day, our my learned colleague, Dr. Ghosh, also explained you regarding this kind of. She also explained the free DNA will be chopped out inside our cells. So to overcome this problem, we have to have some vector that the role of the vector is to protect your external DNA from internal cleavage. So we just took a good copy of DNA because we can synthesize the DNA because we have now the option of PCR. We can insert the DNA into a vector, so we can make a recombinant DNA. This recombinant DNA we can artificially introduce in bacteria. So now the bacteria, upon introduction, this foreign DNA that is better known as artificial transformation will harvard the extra copy of DNA. So this extra copy of DNA will be expressed and the bacteria will show a recombinant or hybrid phenotype. So this is the experiment. This is the basic experiment to modify a simple organism to get a better organism. So I try to skip the overall fundamental things related to these topics. Next slide, sir. Now I just conceptualize in this fashion. We can also prepare insect resistant plant. So we are very much familiar with BT cotton, BT tomato. Just I want to concentrate in this phenomena. So the outcome of BT cotton or outcome of BT tomato is the outcome of genetic engineering in modern world. So normally cotton are sensitive. Hello.
Milon sir, Dr. Edmond had he is in a little bit of hurry, so can you please sum up within a few minutes?
Okay, sir. Just 2 minutes. Okay.
Yes.
So okay. This is the genetic engineering pattern. So we can design the resistant plant from the background of genetic engineering. And next slide, sir. Uh, uh, so this is the next slide. Last couple of months, we this is the this is the genetic therapy. So last couple of months, we are familiar with two things. Another is infection, and another is uh, uh, genetic disorder. So we can overcome from this genetic disorder from genetic engineering. This is known as gene therapy. So that can be two types: somatic therapy or germ-like therapy. And I want to conclude. Sir, last slide, please. I want to conclude my with a thank you to all of my participants and viewers. This is an image of Macron, Great Merkel, because we are running this in a pandemic scenario. So we are not able to deliver our lecture in front of a classroom. So we have to abide by seeing virtual talk. So I want to thank you all the listener, and I also thank you our vice chancellor, our organizer team, my colleagues to help me to share my views. Thank you, sir.
Thank you, Dr. Bonik. I mean,
It
I'm
is
very
a
much
really
sorry.
marathon journey. Sorry.
Yes, no, no. Actually, I mean, if it was as previously scheduled, your talk will be only talk. Then will not have any problem. So I'm extremely sorry, Dr. Boni. That meanwhile I have to, stop you before few slides. It was an extremely good talk. We are enjoying a lot, but unfortunately we are going quite late. Okay,
Oh,
so
thank you, sir.
extremely
Sorry,
sorry.
sorry if I
But
sorry
now
for my.
we have our next speaker, Dr. Edmond Chua. He is from Singapore. Dr. Chua, are you there?
Yeah, hi, hello.
Hello, hello. Yes, yes.
Yes,
This is Rajat Pal this side,
yeah,
and I'm welcoming you on this online course, which is going for the last 5 days. Today is
yes.
the last day, and we are very much fortunate to have you with us. And I think my participants who are actually participating for last so many days, they are going to have a very exciting talk from your side. And I'm just inviting you
Yeah.
to deliver your lecture. And just go ahead. The screen
Sure.
is yours, sir, now.
Thank you
Please.
so much.
Yes.
I'm just sharing my screen. Sorry, is it coming up?
Yes, it is coming, but we cannot see anything.
Oh,
Yes,
right. Okay. Are you able to see it?
yes. Now we can.
Awesome. Great. All
Yes.
right. So before I start, I want to thank the organizers for, organizing this workshop and then inviting me to give this talk on spectral flow cytometry. My name is Edmond. I'm the sales and marketing manager of East Asia for SciTech Biosciences. We are a flow cytometry solutions company, and our specialty is looking at full spectrum flow cytometry, also known as spectral flow cytometry. So a little bit of background about myself. I'm actually used to work as a core manager in running a flow cytometry core manager in Singapore in A-STAR, which is a national research institute. And I used to do a lot of research work into Zika virus in non-human primates, in monkeys, and as well as human clinical trials for vaccines, and as well as for um, some of the disease models that we have, using flow cytometry was one of my main scope. I had a lot of work dealing with a lot of different flow cytometry instruments. I wanted to introduce this new innovative technology that we now call spectral flow cytometry. So a little bit of background of what flow cytometry is before we actually move on into spectral flow cytometry. In the 1980s, when there were only one laser, two colors, that was when flow cytometry was introduced. And looking at that, we had FITC and PE that are still very commonly used now. Fast forward 10 years later to 1990 with two lasers and four colors available for use in flow cytometry. So FITC, PE, PERCP, and APC, which are still a very common combination for researchers all around the world where they are doing four colors experiment. Being able to do four colors is a basic need now. And then, fast forward all the way now where we have more lasers and a lot more colors. We have 5 laser systems that are coming out with 30 different channels and a lot of colors. What you want to notice is that
Dr. Edmond,
yeah, sorry,
please, just sorry to disturb you, but maybe
yeah.
something problem there. Your slides are not visible to the participants. They
Oh,
are
okay,
talking in the chat box. So, can you please,
so
share it once again?
sure. Let me stop and share it again.
Yes.
Uh, all right, is that better?
It has not come yet, actually.
Oh,
Can you please mail me your slides so that I can share?
sure, sure, sure.
Kindly, please.
I'm sending you a link with the download because the file is too big.
Okay, or else once again you can just please try to share, because a few participants were telling that they can, can see.
I see. Okay,
Yeah, so can you please?
sure.
I can see it. So please, participants, just type in the chat box whether it is visible or not. Few of you. Kindly respond so that Dr. Chua will continue. Yes, visible. Yes, sir. Please
Yeah,
come.
visible. All right, great, awesome. So yeah, we were talking about right now in 2015 and beyond, we have instruments that are coming up that have 5 laser systems with multiple different channels and a lot of new colors that have come about ever since the 1980s. So this has been a tremendous growth in the flow cytometry field. You can now detect, 20, 30 markers in one sample, in one cell. That is a great achievement from the 1980s. And what you want to notice with, how flow cytometry has moved across the last few decades is that it has always been looking at a very narrow bandwidth of light to identify a fluorochrome. And so you can see that, in order to identify these fluorochrome, there's a filter or bandpass filter in place to detect it at its emission maximum so that you can get the signal of these fluorochrome. And so flow cytometry, again, how it works in a system is you have a sample, your single cell sample, single cell solution running into the flow cell. It gets excited by the lasers on the instrument, and basically the fluorescence is then detected by different kinds of detectors, each detecting one fluorochrome. And basically, there's an electronic measurement of volts against time. And finally, it would show up as a data point on your FACS plot that you are very familiar with looking at. All right. So that is basically how a flow cytometer works. So the only difference with full spectrum flow cytometry or spectral flow cytometry is that we are changing.
I'm just, please again, I'm disturbing you. Maybe some serious net issues are there. So again, I'm just sharing. Okay,
Okay.
Edmond, please just. It's a very problematic going on. Maybe somehow. So I have just take it in my laptop. So I'm just sharing it. Okay.
Okay.
Let
Sure.
me item to review.
Yeah. So you can just follow my my slides and move on to the next page.
Yes. Yes. Sure.
Yeah.
Sure.
All right. So is it open? Should I continue?
You just stop sharing, please, so that I can share.
I see. Okay.
Now, is it visible?
Yes. Now I can see it.
Okay. Okay. So you just go ahead.
Okay. Yeah. So if you enlarge the screen in presentation mode. Yeah. Great. So you can fast forward a couple of clicks.
Hello.
Yeah.
Yeah. Do I have to do something?
Yeah. You are you able to move to the next slide? Yeah. So there's an animation there, and then to move on to the next slide. Yeah, and then next slide. Yeah, here. So basically, when we are looking at conventional or all kinds of flow cytometers, one of the things that we have to deal with is that you have looking at the bandpass filter to identify the fluorochrome. And you can see that you are actually only getting information in this very narrow bandwidth or very narrow area, and you are missing out all of these other regions on the side of where the filter is detecting. And so it is akin to like looking at the tip of an iceberg, where you're looking at a really small portion of the information, but you're not really looking at the whole picture. All right. The next slide. Yeah. So spectral flow cytometry is something that is different. Next slide. Thank you. So spectral cytometry is defined as using dispersive optics such as prisms or gratings, and we are dispersing this collected light across a detector array, and that allows the full spectra of each particle to be measured. So as you can see on the diagram on the right here, what we are doing is we are actually not only just using a very narrow detector, but we are detecting across the entire emission of the fluorochrome in order to identify what the fluorochrome looks like. All right, next slide. So I want to reiterate that there is no difference in a conventional cytometer versus a spectral cytometer, except for two elements here, which we have on the right highlighted in red, the dispersing element and the multi-channel light detector. You still have your lasers on the left, exciting your fluorochromes, exciting your labeled cells, and they are then excited, and the signal is then collected via a different way by like looking at the dispersing element and the multi-channel light detector. So we'll take a look at what the dispersing element and multi-channel light detector looks like. Next slide, please. So there are a few different ways that the different systems can actually do light dispersion. There's the way of looking at prism, whereby you shine your light collected through a prism to then split the light beams. We also have diffraction grating, which is where you have a emitter light from the specimen being diffracted through a series of grates. And finally, we have coarse wavelength division multiplexing, also known as CWDM. So all of these are different ways where the light is being dispersed and then being able to detect by the detectors to get the full spectrum information from your fluorochromes. Next slide, please. So we have the multi-channel PMTs, which are commonly used in conventional systems right now. And basically, they are PMT systems which are still used in conventional systems. We have different type of CCDs, which are also used in some of the commercial spectral cytometry systems. And we also have avalanche photodiodes, APDs arrays, which a lot of new manufacturers are moving towards in terms of detecting the signal and the light from your samples. Next slide, please. So there are currently two commercial spectral flow cytometers out in the market. On the left, we have the Sony system, which looks at spectral flow cytometry by looking at prisms. And then the signal, the emission signal is going through a series of prisms to then direct into a 32-channel PMT to look at the spectral data. And we also have on the right Cytek's own spectral flow cytometer, where we have individual emission spectrum from each of the lasers that excite your fluorochrome being collected, and then the information is then stitched onto together to form a signature or how the fluorochrome looks like, kind of like a unique fingerprint to identify the fluorochrome. Next slide, please. Next slide, please. So spectral unmixing is something that is used by all spectral flow cytometers to determine what is the proportion of each reference spectrum that best fits your stain sample. So now, because we are looking at the entire signature of the fluorochrome, we are identifying each fluorochrome by looking at how the signature looks like. So we have over here, for example, on the top left, the FITC signature. So this is how the FITC looks like on the instrument, and so we are identifying this signature or this fingerprint as FITC. And then we have on the bottom left the signature for PE, and this is how it looks like when you run single stain PE. So you can see over here that when you are looking at FITC and PE and you compare across these two signatures, you can see that they are very different across the entire spectrum. So these differences in the patterns, in the up and down motion of the peaks allow the software to spectrally separate out these two dyes. And you can see clearly over here that even when FITC and PE is combined in the top right corner here, by eye, it can be very difficult to see. However, when you overlay them like a plot on the bottom right, you can see that FITC has defining features on the left-hand side here, and then as well as there's a peak here in the median B3 range here, and you can see that in the FITC and PE it incorporates a combination of both these signature. The black line that you see in the plot, it incorporates signatures from both fluorochromes, and that's how you know it's a FITC and PE combined. Next slide, please. So when we are looking at spectral unmixing, we are looking at least squares for solving for the minimum. And so you can think of it as extracting or deconvoluting every component that you have in your multicolor sample until you have nothing left. Next slide. There will be an animation showing you a multicolor sample and how it gets deconvoluted into its individual spectrum, and then it becomes that you have nothing left. It will get broken down into each of the individual signatures. Yeah. Next slide, please. Yeah, you can move on to the next. Yeah. So when we look at the signature of the fluorochrome, what does the signature actually tell us? Next slide. So the signature over here that we get from the system, as you can see on the x-axis here in the plot, we have our channels that we have in the systems, in the detector, in the instrument. So for a 5 laser system on the Cytex instrument, we have 64 different detectors, 16 channels of the UV laser, 16 channels of the violet, 14 channels of the blue, 10 channels of the yellow-green laser, and 8 channels of the red laser. And on our y-axis here is the MFI for each of the detectors. And you can see that when we are looking at this signature, we have a lot of peaks in the different channels. But you can see that in this example, that the UV laser channels on the left here, you can see that there is a peak that is really bright in the UV14 channel. This gives us an idea that this is a UV laser excited dye, and that we are able to identify these as a fingerprint for a UV dye. All right. We can also see that there are peaks in the violet channels, in the blue channels, yellow, green, and red channels. So this fluorochrome is excited by all the 5 lasers in the system, and this gives it a very unique signature or a unique fingerprint to identify this fluorochrome. Yeah. All right. Next slide, please. So each fluorochrome that you actually identify in the instrument must be unique. It can create a spectral signature across the detector array. Oh, sorry, if you could go back one slide. So spectral signatures must be unique in order to be distinguished. So in this case, you can see that BV421 top left here, FITC bottom left, PE on the top right, and APC on the bottom right all look very different in terms of all of their signatures. And that's why you have so many detectors on a system. All of these detectors are meant to bring out the differences and bring out the similarities between these different dyes. You can see that these dyes are very, very different in terms of how it looks on the instrument. So we are identifying that they have very unique fingerprints for these dyes. All right. Next slide, please. And so what does the signature give us information about? We can use the signature to identify whether two dyes can be used in combination. So for example, APC and Alexa Flora 647, these two dyes are not used in conventional cytometers because they are both detected in the same region. And so if you're only using one channel to detect these fluorochromes, you will not be able to identify or separate out these two dyes. But when we look at APC and Alexa Flora 647 on the left-hand side here, you can see that the circled regions are very different in terms of the signature. So it means that we can run these two dyes or these two fluorochromes in the same combination, in the same panel, in the same sample, and we can still separate out these two dyes. An example on the right here where we have FitC and Alexa Flora 488. When you look at the signature, they are very similar. There's no differences. There's a lot of similarity. So it would mean that these two dyes are too similar for us to separate out. There is no differences in the signature, and so it's very difficult to separate them. So we cannot use these dyes in combination. All right. Next slide, please. Yeah, so when we overlay the two slides, the two signatures for the FitC and Alexa Flora 488, you can see that they are almost overlapping, very, very similar. And so it's very, very difficult to actually pull apart these two dyes because they are too similar. All right, next slide, please. So we can also use the signature to evaluate spread. In the past, when you are trying to evaluate spread, you always have to run your samples before you can actually look at the spreading of your populations, and then you can make changes there. However, now, because we are looking at the full spectrum signature, we can already determine the spread that will be introduced into your sample by looking at the single stain controls. So by identifying that, for example, in this case here on the top left, we have PE-Sci5 and as well as APC, the spectrum signature. And you can already see that the regions where they are circled in red, box up in red, is where you will see the most spread because that is where it has a high degree of similarity. So next slide, please. And so when you look at the normalized spectrum, that's what you expect. You can see that there is a little bit of similarity on the right-hand side here in the box up red square. That's where you will expect a lot of spread. So is that what we see in the data? Next slide, please. Yeah. So based on the spectrum, this is what we would expect to see, that in the red channels, we are going to see a lot of spreading coming through into the from APC into the PE-Sci5. And so if we look at the data, next click, you can see that that is what we expect over here. If you look at PE-Sci5 versus APC, there's a lot more spread of PE-Sci5 into APC, but not in the other direction. So there's less spread from APC into PE-Sci5, but more spread from PE-Sci5 into APC. And that is what we will see from the data as well. So we can already determine or already evaluate the spread that we will see from our single stain controls. Next slide, please. So another thing with looking at full spectrum would be also to be able to check on the reagent integrity. Next slide. One of the things we can do is, because we already know how the signature or how the fingerprint of each single stain looks like, we already have a database of it. We can actually check and see how your single stains or your antibodies are performing, whether they have degraded, whether they have changed their properties before you stain them on your samples. And that helps you to save your sample, helps you to save your reagents as well, and help you to save your effort in running the experiment. So next slide. And so an example like this where Q.605 looks very bad tells us that there is something wrong with either the antibody or the sample itself. So that gives you an indication of how whether you can still continue using this reagent. All right. Next slide. Yeah. Next. I'll skip this one. Next slide. Yeah. Yeah. So it also can do quality checks as well. Next slide, please. So we can look at the single stain controls, and we can actually see if your single stain controls are done correctly as well. So for example, over here, we can see on the top right that we expect the signature for E-fluoro-450 to look like this, the signature. However, when we look at how the signature actually looks, it looks like it's contaminated with APC, which can be an accident sometimes where, you have wells or tubes that are next to each other and you accidentally added in the wrong antibody, and that can happen, and we can immediately catch that here when we are looking at these kind of data. Next slide, please. Next slide. I'll skip this ones. So another feature of full spectrum flow cytometry or spectral flow is that we can do autofluorescence extraction. So a lot of times users run into a lot of problems with high autofluorescence samples, things like tumor samples, things like organ samples from the different organs, cell lines that have a lot of high background, a lot of autofluorescence. Being able to extract the autofluorescence would help in getting their resolution improved and getting their data much better. Next slide, please. So different types of cells have different patterns and levels of autofluorescence. As you can see here, splenocytes are pretty clean. Lung cells are very autofluorescent. They have a lot of background, which you would expect from the organ. And then you have E cells, which are also highly autofluorescent, a lot of background, very difficult to get good signal or resolution in flow cytometer. Next slide, please. Yeah. So some of the advantages of spectral cytometry. Next slide. Is that we are able to collect more light. So for example, in this case, you have the PE spectrum, the full signature over here of how PE looks like. And if you are actually just looking at a conventional system, you will be only detecting a very narrow bandwidth of light. Next slide. And so the full spectrum ability also enables us to use highly overlapping dyes. An example here would be the example we've shown just now with APC and Alexa-fluoro-647, where when you look at the signature of APC and AF647 over here on the bottom left, you can see very clearly that they are very different in the signature, where they are very unique areas and very unique differences. And so that means that we can actually run these two dyes in combination. So say, for example, CD57 Alexa-fluoro-647 and CD8 APC on the right over here, you can see that we are able to run them together in the same panel to look at co-expression populations. All right, next slide, please. So another feature like we talked is the autofluorescence extraction. This is the unstained sample on the top left here from the E cells. And then we have the CFP in the next bottom one. And you can see that it looks very, very similar. And that is why when you have no autofluorescence extracted, like your example here on the right, you can see that your unstained or your negative cells will actually be wrongly identified as a CFP positive cells. That is because it is very, very similar, looking very similar with the unstained versus your CFP single stain cells. However, if you extract the autofluorescence, you will now be able to see that the population, the negative population, is back down to 0. I'm not sure whether you can see very clearly, but the red arrows over here show us that they are pointed at 0 points. So your negative population is centered around 0 again and not showing a false positive. and the resolution looks great. You have better separation between your positive and your negative population. And then next slide, please. So I want to show you a 31 color marker that has been run on the Cytec Aurora, where we have not, we can run so many different markers, so many different colors on a 5 laser system without sacrificing the resolution. It's a highly overlapping panel where we have things like BV421 and eFluor450 in the violet. And then we also have APC and AF647, like the red laser excited fluorescence, as well as things like PerCP Sci5.5 and PerCP Fluoro710 in the blue laser excited fluorophore. Yeah, next slide, please. So when we are looking at 31 colors, we're not really looking at a very general overview, but we are looking at the deep subsets of what you can find in human blood cells. So not just T cells, B cells, but really digging very deep into how B cells and T cells look like on the in your sample. Yeah, next slide. So as you can see over here, it's a very busy slide, but I would just want to draw your attention to the center of the slide where we have the CD4 and CD8 expression. You can see that the CD4 and CD8 are very clearly separated. And then when you get on the subsets of these CD4s and CD8s, you can see that the positive and the negative populations are clearly separated. The negative population is tightly clustered around 0. And you can also isolate in the bottom right here, Treg cells using CD25 and CD127. So you can see that there is you can look at rare subsets of cells, and from the rare subsets of cells, you can look at the subsets of these rare subsets. So it gives you access to a lot of data, and then you can isolate the different subsets of B cells, NK cells, and monocytes as well. Next slide, please. So I'm going to skip this. So next few slides, please. Next. 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 Next one. Yeah, so so you have seen some of the signatures just now, but what I want to say is that we have helped our users kind of like made it easier for them by recording these signatures and showing them these signatures to help them to set up the instrument so that they don't have to rerun their single stain controls every single time. So you can set up your single stain controls once and be able to use the single stain controls without creating them again. So you can store them in a library because the fingerprint for the fluorochrome does not change from one day to another. It's always going to be the same. So you can store the reference control or the single stain control in the library, and you can reuse them as many times as you would like in a certain time frame. So next slide, please. So we tested out how it would look like in multiple different sites. The data, we look at cross-site assay performance, looking at the different same panel in the different sites, and you can see that it's very reproducible. You get the same kind of unmixing, being able to unmix and get your rare subsets of cells like Tregs, and then looking at your CD4s and CD8s as well. Next slide, please. And this is a 22 color data that we have done across standardized unit, just to show you that, across multiple different units, we are getting the same kind of capability, the same kind of reproducibility when we are doing the data. And, we don't have to, you can use the controls that are stored in the instrument to reuse them so that you don't have to prepare single stain controls each time you run your experiment. Next slide, please. So in summary, I want to just show you that the full spectrum analysis can help you to predict dye compatibility, and it offers you great flexibility in the number of colors that you can choose in your flow experiment, and the ability to give you high quality, high parameter data. And we also offer the standardization of instrument across multiple sites. So it's great for people who are doing multiple sites, different kinds of clinical trials that are across different parts of the country or different parts of the world. Yeah. Next slide. And I think that's the last one. Yeah. So I want to thank everyone for being on the call, and just the organizers for inviting me to give a talk on and to be able to introduce spectral flow cytometry to all of you. I have my email on the first slide. If you had a chance to take that down, otherwise I'll type it in the chat box, and please reach out to me if you have any questions. Yeah, in the first slide. Yes, that's right. Thank you so much. So my email is there. If you have any questions regarding spectral flow cytometry or regarding the presentation that I've just given, please feel free to reach out to me with my email over there. Thank you.
Okay. Um, it's a very nice talk, and I mean, nothing I can say. It's a wonderful journey. I mean, it's a very good knowledge we have gathered that how the flow cytometry actually works. This is actually the students, for the participants who are very young. For them, I'm just telling you, in our this whole program we have merged two things. Okay, first is a very basic what we people have talked, and you can see that from the industry personals we have, I mean, called them to let you know how our knowledge actually has been used in the industry. Okay, so the very young participants may be thinking that these are too tough, these are not we are getting. Actually not like that, we just wanted to share you, or we just wanted to let you know that how our knowledge actually we are, I mean, used in the industry. So you can see in the very first day Dr. Mojumdar told you how we can use the bioinformatics tools in the university. Yesterday Mr. Ghosh told you how, I mean, our microbiological and biochemical characterization techniques we can use in the water purification industry. And today, Dr. Edmond Chua told you how the I mean, cellular morphology and the reaction with the different kind of dyes we can use for the differentiation among the different types of cells. So thanks a lot, Dr. Chua. I mean, you, in spite of your busy schedule, you have got the time for us, and you spent a lot of time. And I just, I mean, beg a pardon for you that for little bit of delay. There are so many, I mean, as we all are doing in the virtual platform, in the online system, so we have to, I mean, we have to depend on this system, and this is nothing on our hands. So I'm extremely sorry for that, for the delay. And thanks a lot from in the behalf of the Department of Microbiology and Biotechnology, Sister Nivedita University. I am once again thanking you,
Thank
Dr.
you. Thank you.
Chua.
Thank you so much.
Yes. So with Dr. Chua's talk, we are actually at the end of our session. It was a very nice journey. I have gone through a very nice journey and a very good experience with all of you. I must say, it is very, I mean, with a very big pleasure and with very happiness, I'm sharing you that we have already 450 participants for the last few days. So it is a very successful journey. And for the very first time, we were trying to, I mean, organize this kind of webinar, and the success makes us overwhelmed. And we all are very much grateful and thankful to all the participants to be with us, my dear students, my university students, and the students apart from my universities all over the India. And I hope that we will come back soon with our new program. And Dr. Thakur Chaudhary, may I request you to share the feedback link in the chat box, please, so that the students can, or the participants can.
Hello, Rajat sir. I have already shared the feedback link. Students can see the feedback link in the chat box, both in the meeting as well as in the YouTube chat.
Thank you. Thanks a lot, Dr. Thakur Chaudhary. As the head of the department, at the end of the session, 5-day online program. I'm feeling a little bit of emotional that I had spent 5 days with all of you guys, and at the end of the day, I am again hoping when I will see you all together in a single platform. And at the end, I would like to request you all that please do visit our beautiful campus of Sister Nivedita University at Kolkata. If you do visit, whoever outside the Calcutta are participating this webinar, whoever they are, if you anytime visit Calcutta, then please let us know, and please do take a visit to our beautiful campus of Sister Nivedita University. Okay, the Department of Biotechnology and Microbiology always welcomes you at the Sister Nivedita University. And add a vote of thanks. I would like to thank first. I would like to thank my honorable Vice Chancellor, by whose inspiration actually we have. I mean, we can have this whole session. Without his inspiration and without his planning, we cannot do anything. So it's a very, big thanks to our Vice Chancellor, Sir Professor Dhruv Chattopadhyay. I want to thank all my faculty colleagues, Professor Shomshpur Thakur Chaudhary, Professor Atri Ghosh, Professor Pratima Calcutta and Professor Milon Boni. I'm also thankful to my guest speakers, Mr. Gourab Ghosh from Mark Life Science, Dr. Mohit Mojumdar from Fine Biotech, and Dr. Edmond Chua from SciTech. So everyone, I am very much thankful to. And lastly, I would like to thank all my participants who have been with us for last 5 days. And this is everything about the session. And with the hope of the next program, I am concluding this session. And I would like to all of you to please stand up for our national anthem. We all are going to sing. Please unmute yourself. Whoever from India, kindly stand up, as it is our tradition. We all are going to end up our session with our national anthem. Please kindly stand with us. Make yourself unmuted. Yes, start. जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड़ उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्छल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मागे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे. Thanks a lot. Thank you, everyone. So, at the last, I am concluding the session. Please do fill up the feedback form, and with due permission from our faculty members, Dr. Thakur Chaudhary, Dr. Atri Ghosh, Dr. Fatima Kalkatawal, and Dr. Milon Boni, I am ending our session here. Thanks a
Thank
lot.
you. Thank you, sir.